Welcome to Life in Biology. I'm Dr. Joel Graff. Some students in my class seem to think that my 40-minute overview of our research is somewhat overwhelming, so what I decided to do was to try to summarize my lab's research on one single slide. And so that's what we have here. I have the flow of our work broken down into different phases: phase 1, phase 2, and phase 3. And there's some arrows showing some workflow options as you work your way through. So the number one step for working with genes in my, or potential antiviral proteins in my lab, is that we order synthetic human genes. Currently my lab has about 40. And with those synthetic human genes, we start doing a bunch of cloning. We do both recombination, subcloning, and regular PCR restriction digest type cloning as well. So we do a variety of different cloning options. Once we have our genes of interest into a core set of plasmids, these plasmids are designed to be useful in bacteria model systems, fungal systems such as yeast-2 hybrid, and mammalian cells. They also can be used in vitro reactions as well. So there's a lot of flexibility for studying these proteins using this core set of plasmids. So when we take these human genes and the students clone them into this core set of plasmids, what they've done is created a toolkit to study these antiviral proteins. The plasmids, once a gene is in a plasmid, we can start thinking about doing some site-directed mutagenesis, which was not talked about in the long version of my research summary in the separate video. But in this case, we can think about doing site-directed mutagenesis, where we can go in and change the DNA sequence of the gene that we're studying so that perhaps it loses its enzymatic function. For example, we study ring domain proteins, which a ring protein is ring domain has a bunch of cysteine and histidine residues that are used to coordinate the binding of zinc, and it's important that these cysteines and histidine residues remain intact so that they are able to fold properly while binding to the zinc. And if we change some of those cysteines and histidines, we essentially knock out the functional domain of our enzyme. Other things we could do is there's sites like phosphosite where we can identify places on our protein that are amino acid residues on our protein of interest that have been shown mostly through massive mass spectrometry reactions, which different amino acids are modified with phosphorylation or acetylation or ubiquitination or any other post-translational modified, but those are the three most common. And we can go in and we can change the amino acids. For example, if one of our proteins is phosphorylated on a specific serine residue, we can go in and change that serine residue to an alanine so that the protein will act as if it's not phosphorylated, or we can mimic the phosphorylation by substituting in an amino acid in place of serine that has a negative charge, like glutamic acid. And then finally, we can look into human genetic variants. A lot of human genomes have been sequenced at this point, and we have just one human sequence cloned into our genes, or into our plasmids, but we could look to see what are some other common variants or some disease relevant variants, and we could make those changes to the amino acid sequence of our proteins so that we could test maybe those genetic disease associated variants. So whenever we do that site-directed mutagenesis, we design one set of PCR primers, and that can go back and we can make changes to all of our different plasmids where we have that gene of interest in it. After phase 2 is phase 3, and we have two large areas of research going on in my lab. One is we're trying to study protein-protein interactions. Our proteins are E3 ubiquitin ligases, so we have a hypothesis that if we can understand what protein our E3 ligases interact with, we would be able to assume that that protein may be targeted for degradation or targeted for activation through that ubiquitination. And so we have a number of protein-protein interaction assays that we can use to identify and then also confirm any particular protein-protein interaction. And finally, we have a bunch of functional assays. So those plasmids where we can overexpress the gene of interest in human cells, we can test whether having an abnormally high level of the protein of interest can change transcription factor activity, increase or reduce autophagy. Cell cycle might be impacted by our proteins, and then we can also look at apoptosis and cell death to see if our proteins of interest might cause those to be over or underexpressed. On all of these cases, we kind of are approaching these functions from the viewpoint of how it would, what impact it would have on a virus infection in a human cell. And so that's why we chose these particular assays to understand potentially how our antiviral proteins are working. So that's instead of a 40-minute explanation of our research, that's a nice 6.5-minute explanation, just one slide. If you have any questions, go ahead and post them in the comments, like and subscribe, or not.